Action potentials are brief electrical changes in the neuron that occur when the neuron is stimulated. They are created at the axon when the membrane voltage reaches threshold. The action potential ultimately causes the neuron to release neurotransmitters at the synapse. They occur along the length of the axon, ending at the axon terminals, spreading like a wave as they move. The movements of sodium and potassium across the membrane create the action potential. At rest, sodium is found at high concentrations outside the neuron, while potassium is found at high concentrations inside of the neuron. These concentration differences are established due to the action of the sodium-potassium pumps. A stimulus will cause the opening of voltage-gated sodium channels at the beginning of the axon near the neuron soma, allowing sodium to diffuse into the neuron axon at that region. Sodium creates a depolarization and the membrane voltage moves from minus 70 millivolts, which is at rest, to a maximum of plus 30 millivolts. Voltage-gated sodium channels become briefly inhibited after that, so for a brief time, another depolarization cannot occur. During the depolarization, the sodium channels close before the axon region can exceed plus 30 millivolts. The depolarization causes voltage-gated potassium channels to open, allowing potassium to diffuse out of the neuron. Movement of potassium causes the voltage to drop from plus 30 millivolts towards minus 80 millivolts as potassium channels begin to close. This drop in voltage towards the resting membrane voltage is called repolarization. For a brief moment, that is about a few milliseconds, the membrane voltage reaches minus 80 millivolts, which is called hyperpolarization. It occurs as voltage-gated potassium channels begin to close and some potassium continues to diffuse out as they close. Graphing the voltage change during the action potential, we see the following events. During the depolarization phase, sodium enters the neuron axon region, causing the voltage to rise. After that, the voltage-gated sodium channels close and are briefly inhibited, while the voltage-gated potassium channels open, causing potassium to diffuse out of the axon region and dropping the voltage back towards the resting membrane voltage. This dip is called the repolarization phase. The hyperpolarization phase is caused by the continued diffusion of potassium out of the axon region while the voltage-gated potassium channels are closing. The action potential regenerates at sequential segments down the axon. The action potential in one region creates a new action potential in an adjacent region. In doing so, the action potential moves down the axon like a wave, and this wave-like spread is called propagation. When the action potential reaches the axon terminal, it stimulates the release of neurotransmitters into the synapse. Myelin, which wraps the axon of some neurons, allows the action potential to propagate further before it has to be regenerated, and this results in a greater speed of the action potential traveling down the length of the axon.